Hello, dear students of the first secondary grade, and welcome to a new episode of our program, Bank of Knowledge. Our program is actually new, and we present to you all the curricula that you study. We also present to you revisions. So I hope that you follow us on our program. And on this episode, we're going to have a lesson of biology. Actually, it's the first lesson. We're going to revise the first part of lesson 1, and we're going to move on to the second part and study Mendel's experiments on pea plant with our teacher, Miss فيوليت خيري, senior teacher of biology. حلقتنا لطلاب الصف الأول الثانوي حلقة بيولوجي، وبرحب بكم جميعا وبرحب بحضرتك Miss فيوليت. Welcome to our program.
Hello.
So let's first start by revising the first part of lesson 1. So what are you gonna discuss here?
Hello, my dear students of first secondary grades. Today we are going to revise the important points that you have studied last time in biology in the first lesson. So you have to remember some important points like the chromosomes. What are the chromosomes? These bodies that are located inside the nucleus, and the chromosomes are present in the nucleus in pairs, and these pairs are called homologous chromosomes. So these homologous chromosomes means that two chromosomes have the same size, containing the same type of genes, the same number, arranged on the same way on these chromosomes. Actually, the homologous chromosomes, one comes from the male parent and the other one comes from the female parent in the somatic cell. So the chromosomes carry genes. Of course, you studied the chromosomal theory, and to remember them, to remember the points of the chromosomal theory, the chromosomes are found in the somatic cells in pairs, as we said. The reproductive cell, which is gamete, contains half the number of the chromosomes, n, and this number is reduced to the half because or during the meiosis division. So during the meiosis division, the 2n becomes n in the produced cell. Each pair of chromosomes behave independently at its transmission into gametes. So during the formation of gametes, the homologous chromosomes behave independently from each other. One comes or goes to the gamete, and the other goes to the other gamete. During fertilization, the gametes fuse together, the male gamete and the female gamete fuse together and produce the new cell of the first of the next individual, which contains the cell contains 2n. So the number of chromosomes is restored to the 2n as the original cell in the somatic cells of the individual. Genes are located on the chromosomes, so each chromosome contains many genes. So the number of chromosomes, of course, less than the number of genes. The genes number may reach hundreds on each chromosome. And now we are going to start the Mendel's experiments.
So now we're going to move to the second part of lesson 1, chromosomes and genetic information, and we're going to start with Mendel's experiments. So what do you have to say here?
Mendel actually was the first scientist to put the foundation of the genetic science.
So we can call him a pioneer in this
pioneer,
field.
Yes. He studied the pea plants. He was cultivating them, and he was very near or close to the pea plants, watching the characteristics of them, and he could tell us about the principles of genetic materials or genetic information that are transmitted from one generation to another generation. Mendel studied 7 pairs of allelomorphic characters represented by 7 pairs of genes. So each pair of genes locate on 1 pair of homologous chromosome. Each pair of these genes locate on 1 pair of homologous chromosomes, like this picture. And these 7 characteristics in the pea plant, like this. The seed shape, it could be round or wrinkled. The seed color, it could be yellow or green. The pea shape or the pod shape, it could be inflattened or constricted. The pod color could be green or yellow. The flower color could be purple or white. The flower location could be axial, comes out at a side of the stem, or at the end or the top of the stem, terminal. The pea plant height could be tall or short. So as you notice here, for each character, there are 2 shapes or 2 phenotypes, 2 morphological shapes or characters. So this is what we call the allelomorphic characters. In Mendel's experiment, we could notice from this picture that he pollinated 1 flower, the pollens from 1 flower to the other one, to make cross-pollination. And after fertilization, he and the formation of seeds, of course, he cultivated the seeds to show the new generation of the plant. He transferred pollen from a pure white flower to the carpel of a pure purple flower. Then he got all plants with purple color. This is one of his experiments. Like this. When he cross-pollinated a purple flower with white flower, he found that all the first generation were purple, as you see in this picture. And afterwards, he let the first generation to pollinate themselves or have self-pollination, and he got to the second generation, some of them purple and some of them white. Actually, when he calculated the number of purple plants, purple flowered plants, he found them about 75%, and 25% was white. So the ratio here, as we see, we call them 3 purple to 1 white. To understand what he did or what he deduced from his experiments, actually at Mendel's time, he couldn't explain this result perfectly or accurately. But afterwards, when the scientists discovered the DNA and the chromosomes and the genes, we can explain them, explain the experiments in a good way. So let us do it in another way. He pollinated, cross-pollinated the purple flowered plants with white flowered plants, and these are the parents. And because each character is represented by 1 pair of genes, so for each character we have to think about 2 genes. And because Mendel found in the first generation all individuals were purple flowered plants, so he called the character which appeared in the first generation dominant. So the dominant character is the character which appears in the first generation. And referring to the chromosomal theory, we have to put letters or put symbols for each character. For each character, two genes or one pair of genes, so we have to use letters. What are these letters? We use the first letter in the dominant character to express the character. We put these letters in capital way to represent the dominant, and we put the same letter in small to represent the recessive character. And of course we have two genes, so we have to put two letters, one for each gene, capital P, and for the white character, small p, small p. And this is the case of the parents, the genotype of the parents. But the parents need to form the gametes. The gametes in the plants are pollen grains, which are the male gametes, and ova for the female gametes. And of course, as you remember, that the gamete is a cell, reproductive cell, contains half the number of chromosomes. So we have to find the half of these chromosomes. We take just one in the gamete, as we said, during the gamete formation. And from here, we take another one. So if these are the pollen grains contain half the number of chromosomes. So we are speaking about one pair of chromosomes, so we take one of them. And of course, they are the same, so we take just one. And here is the same. We take just one in the gamete. So the cells here are 2n, but the cells here in the gametes are n. After fertilization, the two gametes fuse together to produce the new individuals, capital P, small p. So since the capital P is present here, so the individual expresses the dominant character. And the recessive gene doesn't appear, has no effect in the appearance of this character. We can make them in another way. We can say that the capital P, which is the first parent, small p, second parent, and this parent forms gametes, and the gamete here is P, capital P, and the gamete in this case is small p. By crossing them together, capital P, small p, capital P, small p, all of them will be capital P, small p. So all individuals become purple. So 100% of the first generation is dominant or carries the dominant character. All of them are purple. This is regarding to the...
Can you just adjust the paper a bit in الهواء actually? Yeah.
Okay, so this is what we got from the first type of first experiment that Mendel carried out. Interpretation of Mendel's laws according to the chromosomal theory. How can we explain Mendel's results in the regard to the chromosomal theory? This is what we have said. During the gamete formation, the homologous chromosomes separate, like this picture. And you can notice that we start with a certain number of chromosomes 4 chromosomes, and we end up with 4 cells, and each one contains half the number of chromosomes. These are the gametes. The gametes in case of the plant are pollen grains, male gametes, and ova, female gametes. But during fertilization, the gamete containing half the number of chromosomes in each parent will fuse together to restore the original number of the somatic cell. So we have here some important terms that we are going to use quite often in the next lessons. The dominant character. What is the dominant character? Here is the dominant character. The purple, which appeared in the first generation, even if the gene of white color is present, but the dominant gene for purple color dominates over the white gene or doesn't show its effect. So this is the dominant character. Recessive character, it must be always pure because it cannot appear unless the dominant gene is not present. So there must be 2 letters or 2 recessive genes to show the character, the recessive character. And also we have another important term, homozygous. The homozygous means the individual that has the 2 genes are similar, whether they are dominant, capital P, or recessive, small p. So the 2 genes are similar. This is why we call it homozygous. And also we call it pure genes. The heterozygous is the individual that carries 2 different genes or 2 different alleles, the dominant gene and the recessive gene, capital letter and small letter. So we express the heterozygous by 2 different letters, one capital and one small. The phenotype is the description of the character. The phenotype could be the color, the shape, the height of the plant, or any other description of the character that shows up on the individual. The genotype is the letters that we use to express the gene case, whether it is pure, like this, capital T, small t, or hybrid, capital T, small t. So these are the letters that we use to show the genotype. And now we are going to study the other part of our lesson.
We're talking about the laws now, right? So,
yes.
How many laws are there?
We have studied one law which speaks about the segregation of chromosomes.
Yeah.
So as we said, we have one pair of homologous chromosome contains 2 genes, and during the meiosis or gamete formation, the 2 homologous chromosomes separate. So this is the segregation of chromosomes. Right. Each one has its gene, carries its gene, goes to a gamete.
We just got 6 minutes left, so try to sum it up.
So we have another part, this when Mendel studied 2 allelomorphic characters. For example, he chose the color of the seed and the shape of the seed. The color could be yellow as dominant character, green as a recessive character, and the shape could be smooth or wrinkled. Smooth is dominant and wrinkled is recessive. As you see here in this picture, we put the parents. The parents, we represent the homologous chromosomes. One carries capital Y for yellow. Of course, as we said, we chose the the first letter in the dominant character. Capital Y for yellow, small y for green. Capital S for smooth, and small s for wrinkle. During gamete formation, we take one chromosome from each pair. And of course, each chromosome carries its gene. So we have two gametes. One of them carries one capital Y, the other one capital S. So the gamete is capital Y, capital S, and the other gamete small y, small s. After fertilization, we have the new individual carries capital Y, capital S, small s. And we start with these parents to show the second generation. If we let the individuals carry out self-pollination, we will get this table. And this table expresses the gametes of the first individual. We assume it is female, and the second individual, we assume it is the male. So we have capital Y, capital S, capital Y, small s, small y, capital S, small y, capital S for each individual. After crossing them, we will get this result. And you can easily count how many individuals are having yellow seeds and in the same time smooth seeds, and how many yellow with wrinkle seeds, how many green with smooth, and how many green with wrinkled. Because we study two genes or two pairs of genes, so we will have another ratio. By counting these numbers of individuals having these phenotypes, we will find that for yellow and smooth seeds, they are 9. For yellow and wrinkled seeds, they are 3. For green and smooth seeds, they are 3. For green and wrinkled seeds, they are 1. As you notice that the yellow and smooth seeds are the dominant characters, and they represent 9 of them. Yellow, dominant with 1 wrinkled recessive, so they are 3. The green is recessive and smooth is dominant, 3. The both genes are recessive, they are 1. And if we want to refer to the first law of segregation, if we count the number of yellow and green seeds, we will find they are 12 yellow and 4 green. So they represent the same ratio, 3:1. And in terms of the second character, smooth, they are 12, 9, and 3, they are 12. And the wrinkled seeds, they are also 4. So they represent the 3:1 as the first law. So this is the law of independent assortment. Independent assortment means that the homologous chromosomes, they separate independently from each other and produce the new individuals as you see here. So yeah, of course.
Do you want to say something quickly? Or running out of time?
the individuals of the first generation carry the two dominant genes, two dominant characters, sorry, and represent 100%, and the ratio in the second generation, as we saw, 9 to 3 to 3 to 1.
All right, I'd like to thank you very much, Ms. Violet Khayri,
welcome.
Senior Teacher of Biology. Thank you for being with us.
You're welcome.
Thank you for this lesson, and it's a short lesson, of course, a half hour. So I hope you've benefited from this lesson and follow our program on a daily basis. Dear students of the first secondary grade, we've been with you and discussing one of your biology lessons, genetic chromosomes and genetic experiments. We've had a revision on the first part, and we've moved to Mendel's laws and also had some experiments. So I'd like to thank you very much, and until we meet you again, I wish you all the best. Bye-bye.
Bye-bye.